Sure. Thanks so much. Yeah. Do you guys hear me? All right. So today I'm honored to introduce our guest speaker, Dr. Dana Hawley. Dr. Hawley received her PhD from Cornell University in ecology and evolutionary biology, and today is a professor of biological sciences at Virginia Tech. There she investigates the ecological and evolutionary mechanisms that underlie host susceptibility, pathogen virulence, and transmission. Her main projects focus on social behavior and disease, evolution of pathogen virulence, and causes and consequences of phenotypic immune evasion. Today, her talk is entitled, Why Bite the Hand That Feeds You? An Integrative Approach to Studying Virulence Evolution in a Songbird. Welcome, Dr. Hawley.
Thank you so much. It's a pleasure to be here, and I want to see if how my sharing works. So are you seeing my presentation mode, or?
We see everything, including next slide and notes.
Okay, let me switch that. I have two monitors and I can never tell which one is actually being shared. Is that better?
That one looks good.
And also, before we get started, do you prefer questions at the end, or are you fine with people raising their hand during the talk?
I'm fine either way. So if someone wants to jump in with a question, go for it.
Sounds good. Thanks.
Great. Thank you. Well, thanks everyone for being here virtually, and I'm looking forward to hopefully being able to interact with many of you in person in June, knock on wood. So thanks for giving me the opportunity to at least interact with some of you today virtually. So before I dive into the talk, I just want to acknowledge that there were a large number of folks from my lab, past and present, that contributed to the work. And even though I will kind of give a shout out to some people when I get to their particular contributions they led, behind the scenes are a lot of other people that deserve acknowledgement for making sure that the work happens in so many different ways. Let's see, not able to, there we go. Okay, so pathogens by definition cause at least some harm to their hosts, but there is substantial variation in the degree of that harm, which I'm going to term virulence. And I'm using the kind of ecological definition of virulence here, which is the reduction in host fitness due to pathogen infection. So from kind of a conservation and a public health perspective, we're of course very interested in what sort of ecological and evolutionary factors underlie pathogen strains or taxa that have high virulence. Now, from a very broad perspective, we can think of three sort of categories of factors that determine the amount of harm that a host will incur from an infection. So the first one we probably all think of are pathogen characteristics. And in fact, we typically study and quantify virulence as a pathogen inherent or specific trait. So we hear a lot about, Omicron these days, and is Omicron less virulent than other variants of SARS-CoV-2? And we know in many well-studied wildlife systems, like West Nile virus, for example, that distinct strains of virus or other types of pathogens can cause very different levels of virulence. But we also know that characteristics of the host matter in terms of the amount of harm incurred from infection. So to use the same kind of West Nile example, if we give distinct songbird species the same strain of West Nile virus, we will see a lot less mortality in something like an American robin versus a crow, typically. We also see this kind of variation within a species. So to go back to the SARS-CoV-2 example, we know that there are a lot of factors within humans that determine the likelihood of mortality, including age. And then finally, there are effects of the environment. So it doesn't just matter what that particular host and pathogen characteristics are, but also the environment in which that interaction takes place. So my lab has done some work and continues to do some work in this area too. I'm not going to have time to talk about it today, but we most recently have found that if we house birds at distinct ambient temperatures and give them the same strain of pathogen, we see very distinct degrees of virulence experienced. So you'll probably recognize these kind of three broad characteristics as the 3 points of the disease, or often called epidemiological triangle. So this framework nicely captures the idea that virulence, while it's important to study as sort of a pathogen-specific trait, is very much an outcome of all of these kind of interacting factors. Today, I'm only going to focus on the pathogen and the host, and that's because I'm going to be taking kind of an evolutionary perspective today, and these are the 2 parts of the triangle that are able to evolve. And so I want to start out thinking about a very basic question, which is somewhat easy to answer, and that is, what is the optimal virulence for a host? So if you think about this, you probably very quickly will come to the answer that the optimal virulence for a host is really always 0, or as close to 0 as possible, right? It's if we define virulence as a negative effect on host fitness, then of course a host would want no virulence at all. And so we often think of hosts not really having control over virulence, particularly when we quantify that as a pathogen-specific trait. But when we think about virulence in sort of the disease triangle context, we can also think of hosts as being quantifiable in their kind of contributions to virulence, so to speak. So the way that I'm going to kind of consider that is in the context of tolerance, which Tom Little and others have sort of described as almost the kind of host contribution to virulence. And tolerance is a host response that we think about as minimizing the per pathogen fitness impacts of infection. So we can also define it sort of as minimizing the virulence associated with a given pathogen load. So we measure tolerance using essentially the relationship between pathogen load and the fitness impacts of infection here on the y-axis, which are almost always positive. So in a typical pathogen system, we see that individuals that harbor higher loads of pathogen are going to have higher fitness impacts. But we might see variation in the slope of that line, where you may have some individuals that are relatively less tolerant, or some populations, for example, and others that are relatively more tolerant, meaning that they can harbor quite high loads of the same pathogen, but not experience the same degree of sort of fitness impacts here on the y-axis. Get my sort of, I don't know how to get the pointer here. In any case, so a sort of well-known example of a potentially tolerant host is typhoid Mary, who we know had high levels of the bacteria that causes typhoid, but showed no or relatively few kind of apparent effects of infection. So of course, tolerance is not the only way that a host can avoid virulence. Of course, hosts can also avoid getting infected altogether, or they can resist pathogens once encountered. And I'm not going to talk so much about resistance today, but resistance is the more commonly studied host response, which is just preventing pathogen growth or rapidly eliminating it from the body once it colonizes. And as context, there are several examples of resistance sort of evolving very rapidly, because we know that hosts, of course, want to minimize those fitness impacts of infection. So one of the most notable examples comes from Australian rabbits, where Mycoplasma virus was introduced in the 50s, and within several decades, the rabbits there have evolved high degrees of resistance to this virus. So the conclusion from this kind of short snippet on hosts is simply that there's different ways that hosts may go about minimizing virulence. I'm going to focus on tolerance later in the talk, but optimal virulence for a host is always 0. Now it gets a little trickier when we think about what the optimal virulence for a pathogen should be. And historically, the thinking was that actually hosts and pathogens sort of align in their optimal virulence. And the idea here is simply that, pathogens by definition, of course, depend on their host for their own fitness. And so ultimately, all host pathogen interactions should eventually evolve to be close to mutualisms. And the idea of these kind of highly virulent pathogens was often explained by the fact that they just simply haven't had enough kind of evolutionary time to get to that optimum. And so even things like SARS-CoV-2 are often explained as sort of highly virulent simply because they're not yet, they haven't yet reached optimal virulence in the human population. Now that may be true for a number of pathogens, but we also know that there are lots of pathogens that have been around for centuries or millennia in their hosts, and yet are highly, highly virulent. So we have, for example, the entomopathogenic fungi that cause their insect hosts to turn into mushrooms, killing them in the process. We have sleeping sickness that's been around in humans since at least the 14th century, which causes this coma-like state from a trypanosome infection. And so we know that not all virulent host pathogen infections are simply too new. And in fact, for many types of pathogens, optimal virulence can be quite high. And it's easy to imagine for these types of pathogen systems why high virulence may have a benefit for that pathogen. For example, turning your insect host into a mushroom likely helps facilitate dispersal of fungal spores. And causing your human host to go into this coma-like state may facilitate vector feeding by the tsetse fly vector, thus ultimately kind of increasing fitness of the pathogen. The types of pathogens that have been a bit more paradoxical here are the directly transmitted pathogens, because these are the group of pathogens that we expect really rely on their hosts being both alive and mobile to transmit. And so we generally think of virulence as having a strong negative effect on pathogen fitness by essentially limiting opportunities for transmission. So the trade-off model, which I'm sure many of you are very familiar with, helped resolve this paradox by essentially proposing that virulence is sort of inadvertent side effect of the host exploitation or within host pathogen replication that's necessary for the pathogen to get to sufficient levels to be infectious to another host. So in this sense, host exploitation is positively associated with something that's important for pathogen fitness and something that decreases pathogen fitness, creating this trade-off for pathogens. Now, the trade-off model has been criticized in many ways for being kind of overly simplistic, but it's also led to a lot of really important advances and really nice work in empirical systems by Yap and others, showing how the use of this model and these ideas can kind of help us understand virulence evolution more broadly. But we still have a fairly limited number of empirical systems that we've really been able to explore in terms of virulence evolution. So today I'm going to tell you about a songbird system where we've been able to use this natural system to understand some of the factors that contribute to both host and pathogen evolution in this system. So this is a bacterial infection, Mycoplasma gallisepticum, that I'll call MG for the rest of the talk, that jumped from poultry into songbirds and specifically into house finches in the early 1990s. So this is a common and economically important poultry pathogen, and it largely causes kind of respiratory symptoms in poultry, but in house finches, it's primarily a conjunctival pathogen. So the site of infection is the conjunctival sac, and that is the site of spread of the pathogen. And I should note that all of the strains of MG that we are studying in house finches are kind of exclusively circulating in house finches today. So there's not a lot of crosstalk between poultry and house finches at this point. And essentially what we're looking at is a fairly host-specific pathogen of house finches at this point. Host-specific clade, at least, of the pathogen. So house finches are highly human adapted birds that even if you live in the city down in Atlanta, are likely to be in your backyard. If you have a porch light, they love to breed on top of porch lights, as this female is doing. They also love to use bird feeders. And so the fact that they are so human adapted has probably contributed to their commonness in most of the continental US, which makes them a great study system. The pathogen is an interesting one. It's a molecule bacterium. So the Latin name actually reflects the fact that this particular group has no cell wall. So they are neither gram-positive or gram-negative. This group has secondarily lost its cell wall, which makes them a fairly host-dependent pathogen. So they are thought to not be able to survive well in the external environment, and they tend to be fairly host-specific. So how is MG transmitted in house finches? So given this kind of pathogen biology, that would suggest that it's mainly a directly transmitted infection. And that makes a lot of sense from the sort of epidemiology of the spread. But in terms of like how it actually gets from bird to bird, we know that house finches during the times of year that MG is epidemic, which is the non-breeding season, are actually going out of their way to really avoid directly contacting each other. So there is some physical contact during the breeding season, for example, when birds are feeding young or feeding each other as a mated pair. During the non-breeding season, which is when we see these seasonal epidemics, birds are not really directly coming into physical contact. They do have these interactions like this stereotyped aggressive interaction happening here, where one male is trying to displace another off of the feeder port. But that rarely to never results in actual physical contact, and certainly no like direct physical contact between the two birds' conjunctiva. So the majority of transmission in this system is happening through what we call kind of short-term indirect contact, where, for example, an infected bird might be feeding on a bird feeder or another sort of fomite or environmental surface. They're likely depositing some MG onto that feeder port while they're feeding. And we can swab feeders in the wild and detect varying levels of MG. So we know this happens. And we know from lab experiments that that MG that's deposited can be infectious to a naive bird for at least up to 12 hours, although it does rapidly decline in terms of its degree of infectiousness. So we think that most of the transmission is actually happening from these relatively short-term interactions, where if you've watched a bird feeder at all, you'll know that, especially in the wintertime, there's really rapid turnover of individuals kind of rapidly displacing each other. So there's lots of sort of indirect contact happening on the timescales that would result in MG spread. But an important point I want to make is that even though most of this transmission does have this sort of short-term environmental component, MG really is behaving like a directly transmitted pathogen. And by that, I mean, when we model it, we're not modeling an SEIR pathogen because the environmental stage is really short-lived, probably on the order of like minutes to hours in most cases. And so MG is really, from a virulence evolution perspective, still relying heavily on its host in terms of that host being mobile enough to go to a bird feeder, mobile and alive enough to go to a bird feeder and deposit MG just prior to a healthy bird arriving. So the first recognition of this disease, which was made by bird watchers because they saw birds with these swollen eyes, was made in like the Northern Virginia area. And then scientists at the Cornell Lab of Ornithology were able to kind of take advantage of the fact that bird watchers could actually see this disease in their backyards to track its spread in fairly high temporal and spatial resolution. So we kind of know fairly well like how quickly it spread. And we also know, I should say this is sort of how quickly it spread. So through the eastern range of house finches, I won't really get into their history much, but they're introduced in the eastern part of their range. And the pathogen spread within 2 to 3 years through that entire introduced range. Took a little longer to get over to the West Coast, and I'll get to that for the second part of the talk. We also know what effects it had on population sizes. So not only were our bird watchers kind of looking for disease at their feeders, but they also are a great way to measure the population sizes of varying types of birds. And so Hochachka and Don't used this kind of citizen science data to basically determine that up to 60% of the house finch population was lost with the emergence of this pathogen here at year 0 on the y-axis here, or x-axis here. And one of the hallmarks of this system, really, as a experimental system, is that we can measure virulence and disease, or sort of our proxy for virulence, non-invasively. So we don't have to kind of open up an animal or even have them actually get to the point of mortality in order to measure variation in virulence in this system. We can simply examine the severity of conjunctival swelling, and we largely use a scoring system, but we've also done extensive like image J type analyses that all correlate really, really strongly with the very basic kind of subjective scoring system that we use. And we always score blindly to treatment. So we're always doing the scoring in as unbiased a way as possible. We know these scores are a good proxy for the likelihood of host mortality in the wild. So this host on the left here that's showing really severe disease is going to be far more likely to die than this host here on the right. And that's because the mortality in this system is entirely indirect. And I want to show you a quick video to kind of illustrate what these really sick birds look like. This is just somebody's home video on YouTube. So it's not the best footage, but you can just see how lethargic and vulnerable these super sick birds are. And you can imagine that any predator in the area is going to be able to take this individual very easily. So the mortality from infection in this system is actually from predators taking these individuals rather than from the bacterial infection actually directly killing these birds. And we know this because when we infect these individuals in the wild, in captivity, they get this, many of them get this sick, and yet they recover because there are no predators or need to kind of find food in captivity. So it's a nice system in that sense because we can study and measure variation in virulence without actually directly killing birds. So we've done a lot of studies, and just as background, I want to show you some early work where we were just documenting changes in virulence over time as this pathogen emerged. And to do this, we always use hosts that are of the same background, like genetic background, so caught from the same population, and that had no prior exposure to MG. Because here we're really trying to isolate those sort of pathogen, that pathogen component of virulence in particular. So we hold host background constant, and then we looked at isolates from various years after the emergence, both on the East Coast and the West Coast, and it emerged about a decade later on the West Coast. So that's why you'll see the lines kind of starting at a different place on the x-axis here. And our virulence index is really just the average I-score over time during experimental infections of birds in the same kind of background host. So this is showing you data for 9 different isolates. And although you might immediately notice that the Eastern strains have higher kind of average virulence than the Western strains, I'm not actually going to be talking about that pattern today, mostly because all we can do is kind of hand wave about that, although it's an interesting story in itself. But what I want to focus on is the fact that in both regions, virulence has essentially been increasing over time, or the pathogen component of virulence has been increasing. And I know we don't have a ton of strains here, but we have since added more strains and the pattern continues. And we also know from work from an entirely separate group that used entirely separate isolates and strains that they find the same exact pattern in 50 isolates from the Eastern US. So it's always nice when these patterns can be replicated across groups and in isolates. So we think this pattern is pretty solid in that there seems to be this pressure for this directly transmitted pathogen to be increasing in virulence in both cases. And I should have noted that these are actually separate clades. So the Western MG did derive from the Eastern MG, but it is its own sort of phylogenetic clade. And so in a sense, these were sort of two independent, to some degree independent, instances of increases in virulence following emergence. And this really left us wondering, like, what are the factors that are putting pressure on MG to become more virulent, despite the fact that this pathogen should really rely on its host for its transmission. And we know that it's killing off, 60% of house finches out there. And yet it seems to only be sort of increasing over time rather than the opposite, which is what we might expect. Now, I'm only going to have time to tell you about one small piece of this. We've done work on a lot of different aspects of this story, including the host immune system. And we have been trying to approach virulence evolution from what I call kind of an integrative perspective, for lack of a better word, where we're trying to kind of put together pieces of pathogen characteristics, host gene expression and hormone levels, with kind of whole organism responses like fever and behavior, only one of which I'll talk about today, to try to understand the overall consequences for the ecology and the evolution of both the host and the pathogen. I'm only going to have time to tell you kind of two unpublished snippets from the lab today, but one is sort of from the pathogen perspective, and one's a bit more from the host evolution perspective. So I want to start out talking about what host inflammation and behavior can kind of tell us about pathogen transmission and evolution. And to do that, I want to go back to this trade-off framework. And the first question we wanted to ask was simply whether our system essentially fits the assumptions of this trade-off framework. So we asked the simple question of whether we could find like correlations between a proxy of infectiousness and a proxy of virulence in this system. And so to do that, we simply looked first across those 9 strains that I showed you earlier. But here we are looking at how the average pathogen load they produce in the conjunctiva of different birds relates to their average virulence. And we see this strong positive relationship both within the coasts and across the coasts. And again, we find this with other strains as well that we have looked at since. So we know that this system fits the trade-off model in that this kind of metric of host exploitation on the x-axis here is a predictor of virulence. So to some degree, higher virulence is likely to be favored in this system simply because hosts or pathogens cause higher virulence by replicating in hosts enough to be infectious. But one sort of aspect of the sort of trade-off model that's always left me feeling not completely satisfied is this idea that in the trade-off model, we tend to think of as virulence as only a cost to pathogens in terms of its effects on transmission. And we also know that Ewald and others over the years have sort of hypothesized that there may be a lot of reasons why high virulence itself, like above and beyond the high pathogen loads associated with virulence, may have this sort of direct transmission benefit. So this is not a new idea by any means, but it's something that I don't really have a good term for. So for this talk, I'm going to call this a very big mouthful. And I hate to propose new jargon, so I'm very open to suggestions of a better thing to call this. But what I'm calling it for now is spreadability. And spreadability is essentially a load-independent measure of like how good are you at spreading the pathogen load that you have inside of your body. So a host transmission likelihood would be a function of both how much pathogen load they have, but then also how good are they at actually getting that, coughing out that pathogen load or depositing that load they have onto a bird feeder, for example. So one can imagine why virulence might be associated with higher spreadability. For example, infections that are more virulent may cause higher inflammation, and that may make the host cough more and therefore expel more of the pathogen load that they have inside of them. For the house finch system, more virulent infections are associated with more inflammation, more exudate, mucus coming out of those conjunctiva. And therefore, perhaps that facilitates deposition of the pathogen onto bird feeders. There may also be behavioral components of spreadability. This is easiest to imagine with like vector transmitted infections, like the sleeping sickness that I talked about at the very beginning, where virulence may actually facilitate vector feeding on infected hosts. But it may also be the case that for some directly transmitted infections, behaviors associated with high virulence could actually increase transmission. So in the House Finch system, we know that very sick birds often kind of plop themselves on bird feeders and stay there. And whether that actually augments transmission, we don't have a good idea of, but we wanted to kind of consider both possibilities. And we really wanted to address the challenge of like, how can we separate out this load independent process of what we're calling spreadability from just the higher pathogen loads. And what I mean by that is that we've done a lot of experiments just looking at whether virulent strains have a higher transmission likelihood in these captive settings. And they do. We've looked at probably 10 strains at this point. I'm not going to have time to show you those data, but in general, virulent strains lead to significantly higher transmission. But to what extent is that just a function of the higher within host pathogen loads they cause, this higher kind of spreadability component that we wanted to measure, or both? And so to get at this, we wanted to find a way to isolate this particular component of spreadability. And this work was led by a former postdoc, Courtney Thomason, and a former grad student, Matt Aberle. And so what we did to isolate spreadability is we used a inert fluorescent powder that's been used in lots of systems to measure kind of contact rates. But we paired this like with experimental infection to basically be able to measure how experimental infection with strains of different virulence alters the likelihood of an individual to spread equivalent amounts of powder to their flock mates. That was a mouthful, but I hope that makes sense. We're essentially getting at like a load independent, pathogen load independent measure of contact for these infected birds. So to do that, we used these outdoor aviaries to try to get like as close as we could to sort of a naturalistic transmission setting. Obviously, it's not the same as the wild, but it's as close as we can get in these systems. Using these large outdoor systems really limits our sample sizes, but we were able to have 3 different treatments. And the treatment itself was only given to one bird in the flock, called the index bird. And so we had 3 different treatments: the control or sham infection, where they simply get sterile media. And then we had a low virulence strain and a high virulence strain. Now our strain replication is pretty low here, but we use strains that we know from lots and lots of experiments sort of always behave in similar ways and are fairly representative of kind of low versus high virulence in this system. We tracked the behavior of all the birds in our study using pit tags that were attached to their leg bands. And those basically told us anytime a bird was at one of the 2 feeding ports that were available to each flock through these antennae underneath the ports. We also took video data because the pit tags do not tell us what the bird is actually doing. So we wanted to know not only whether infected birds were kind of sitting on the feeder port, but also like how often were they actually pecking at the food and sticking their head in the feeder port. So we did both. And then our methods, so we on day 0 of our experiment, we gave whatever treatment an index bird in each flock was assigned to. So either control or one of those 2 MG strains. And then we waited until day 10 to actually apply the powder and to measure spreadability. And that's because we really wanted to measure spreadability at the kind of peak infectiousness or at a relevant time point, which we know from other studies is around, day 8 to 10 in this system. So at this point, index birds already had some variation in the amount of like conjunctival swelling that they had. And when we applied the powder, we decided to apply it just around the conjunctiva of the birds so that we were really measuring the most relevant kind of spreadability component of how likely is that index bird to deposit powder from its conjunctival region, which is likely most representative of its likelihood of depositing Mg onto a bird feeder and then it being picked up by another bird. So we created kind of these House Finch superheroes, I felt like they looked like. And our main measure was measuring how much powder spread to flock mates. So to look at that, we were looking for kind of traces of powder using a UV light. So this is an index bird, so it's a little bit misleading because this bird has a lot of powder, but in the flock mates, we were seeing more like these fine traces on our gloves. So we were scoring these from 0 to 3 in all the flock mates 24 hours after we applied the powder. And importantly, we actually we hired someone that was totally unfamiliar with the experiment. So they had no idea why we were doing the study to actually take the powder measurements so that we would make sure that those scores were truly blind, not only to treatment, but even to what the question we were asking was. So our main metric of interest here is this y-axis. This is the amount of powder score on the flock mates. So I need to adjust these axes because doesn't really, you can only have a value of 0, 1, or up to 3, but we only got flock mates that went up to this moderate score. We didn't get any bright scores. So these data points represent flock mates in each case, and the virulence treatment on the x-axis here is the index birds treatment. So the flock mates themselves were not treated, but they received powder from these index birds, which hopefully makes sense. So this was sort of our measure of spreadability. And we of course controlled for kind of flock ID because the same index bird had 4 different flock mates associated with it. But we still found a barely significant effect of virulence treatment whereby index birds that were infected with this higher virulent strain were better at spreading the same amount of starting powder to their flock mates than were control birds in particular. And then the low virulent strain fell somewhere in the middle. So this was kind of exciting for us because we felt like we were able to really isolate this kind of spreadability idea. And although our strain replication is low, it points to the possibility that there are these sort of direct benefits to causing high virulence in this system. We were also interested in the mechanism that might be causing this. So we had a lot of data on behavior from our RFID antenna. So the first data I'm showing you, these are feeding bouts that were pulled out in terms of their length in seconds and then transformed. So these were all of the different foraging bouts that each bird did. And here I'm showing you from peak infection, so the 2 days right before we applied the powder. And the important thing to note here is that we see a pattern for the index birds that we don't see with the flock mates, which is good because these are kind of our controls in a way. These birds were not treated at this time, so they should not differ in their feeding bout duration. But for the index birds, we see patterns consistent with what we've seen in other studies that only compare just infected or not, where we see that birds that are infected had feeding bouts of significantly longer average duration. And this pattern kind of goes progressively up with the higher virulent strain. But of course, we don't know, again, what are these birds doing while they're on the feeder? So simply sitting on that feeder port doesn't mean that birds are actually having any contact with that fomite or with another individual who may follow them. So we also looked at a couple of other metrics, and both of those go in the opposite direction. First, I'm going to show you what we call displacement contacts. So displacement contacts are something we estimate by looking at the number of times that in our RFID data, so our antenna read essentially every second who's on the feeder. And when we see the reading go from the pit tag number of one bird to the pit tag number of another within 2 seconds, we consider that as a displacement where one bird essentially came in and displaced the other. And when we look at the number of times that these birds are displaced during that peak infection, we see the opposite pattern, where these birds that are infected with that high virulent strain are actually quickly followed by a healthy bird, significantly less often than these control birds that do not really seem to differ from the low virulence treatment. And again, no pattern for the flock mates, which is what we would sort of expect given that we didn't give them any treatment. Similarly, when we used our video data to look at the number of times those index birds were actually sticking their head into the feeder port and pecking at the food, birds infected with that high virulence strain showed significantly lower peck rates. And again, it's sort of in this stepwise fashion. So these data are all consistent with the idea that these birds infected, and particularly infected with a high virulence strain, are more lethargic, and they're having these longer feeding bouts, which in theory might lead to more sort of contact with a fomite surface, but in reality probably don't, because they're sitting on that feeder port, but they're actually interacting less with that feeder port. So from all of the behavioral data in combination, it doesn't look like behavior is really underlying the higher spreadability from these index birds, but it's a bit difficult for us to tease apart like how these particular behaviors each contribute to spreadability. Right now, we're hypothesizing that the more likely mechanism is simply the differences in inflammation. So these results are not surprising by any means, because this is why we picked these strains in the first place. But we see that the I scores for the index birds varied substantially. So birds infected with the high index strain had higher I scores than those infected with the low index strain. And then of course our control birds had no I score. So it may simply be that something about the degree of inflammation around that conjunctiva was what sort of augmented the powder spreadability onto that feeder, and the ability of the powder to move into flock mates more easily in the flock mates from this high virulence, these high virulence treatment index birds. So we obviously need to increase the number of strains here, but I think these results are suggestive of the possibility that there may be direct benefits to virulence in this system in terms of this spreadability. And this is probably not something that's specific to the House Finch system. It's probably something that happens in a lot of systems where symptoms are correlated with transmission. And so there's been a lot of talk about that with respect to, of course, SARS-CoV-2 as well, in terms of, we need to really understand what the relationship between transmission and symptoms is in order to be able to make predictions about kind of where SARS-CoV-2 is going in terms of its virulence. So what this might mean from a kind of pathogen evolution perspective is that there should be strong selection on MG to really stimulate that host tissue inflammation if that's important for transmission. And so there may be this benefit to high virulence in the system that's not just an inextricable side effect of pathogen replication, but may actually be a direct benefit of kind of stimulating host tissue inflammation. So we were really curious about what kind of counter selection this might cause for hosts. So that brings me to the sort of second snippet that I want to tell you about, and I'm going to have to move quickly on this one. So we were really interested in what sort of selection might be happening on the host. And it's really difficult to do immunology for non-model systems like songbirds because we just have no reagents. So we had to kind of come up with a starting place. And we decided to start with cytokines because we know they're really important signaling molecules for the downstream immune response, and that they're really important in terms of some of these kind of severe overreactivity of the host immune response. So this was work done by a former Fulbright fellow in the lab, along with a former grad student, Dr. Ariel Leon, and they developed a suite of probe-based RT-qPCRs for a number of House Finch cytokine genes. And this represents actually about a year of work just to get this up and running, since we didn't have a House Finch genome. We still don't. And we started out using these assays just to ask a simple question, which was whether birds infected with more virulent strains show stronger cytokine responses in their tissues. very simple kind of descriptive study. I'm just going to whip through this to get to the more interesting stuff. We had three treatments, three time points. We took tissue samples destructively at these 3 different time points. And just to give you the takeaway, we found that these more virulent strains cause higher cytokine expression for almost all of the cytokines we looked at. For here, I'm showing you IL-1 beta and IL-6, which are both particularly IL-1 beta, pro-inflammatory cytokines. And particularly early on in infection, we found this pattern where this more virulent strain causes significantly higher cytokine expression relatively to a less virulent strain. We also found that among individuals, variation in the degree of conjunctival swelling at the time they were destructively sampled correlated quite strongly with their IL-1 beta expression in that tissue. Now that is not a causative relationship, of course. We need to develop CRISPR technology to be able to do that. But it's suggestive of the fact that at least virulence in the system is correlated with host immune responses. And this in itself is not surprising. We know that host inflammatory responses are associated with virulence for tons of pathogens, including those of humans, like COVID-19, where many of our sort of virulent effects of these pathogens are actually from our own host immune responses. What was interesting about this from our perspective is that this suggests that if there is some variation in the degree to which hosts express these inflammatory responses, then hosts should have some potential to evolve and essentially have some kind of control, so to speak, over virulence. And given the strong selection in the system early on, this is just the graph I showed you at the very start, we were very interested in whether there was selection for host tolerance in this system. And we basically used the geographic spread of the pathogen to kind of use a space for time approach to examine this question. So to go back to this way that the pathogen spread, I just want to highlight that when MG kind of made it over to the West Coast, it did so by going across the top of the country and then down the West Coast, such that a lot of the populations in sort of the middle Southwest, like Arizona, were actually among the last to kind of see MG as a population. So we kind of used this spread to ask whether host tolerance has evolved since MG's emergence in House Finches, and whether we could detect sort of immunological signatures of this. And this was a collaborative NSF grant, and the lead institution on this was actually from Dr. Jim Edelman at University of Memphis, and Dr. Rami Dalul, who's an avian immunologist, was also involved in the project. He was originally at Virginia Tech at the time, and now is down in your neck of the woods. So I'm now showing that sort of map of spread a bit more categorically, just to show you the 7 populations that we sampled for this study. And this work was all led by postdoc Dr. Amby Henshin, who was absolutely fantastic and went to all these populations, including house finches in Hawaii that have never seen MG, and was able to catch large numbers of birds, ship them by air back to either Virginia or Memphis, for these kind of common garden studies. So an important thing to note here is that, you know, the best we can really do in a common garden sense is catch these birds as early as possible in their lifespan. So we cannot breed these birds in captivity, but we catch birds that are within a few months of hatching, and we use antibody assays to make sure that these birds as individuals had no prior exposure to MG. So even though they come from populations that vary in their population level exposure, all of the individuals in our study had no prior exposure. We then infected all of these birds from different populations with a sample size of 10 per population per isolate per dose, with 1 of 2 MG isolates or strains. So we used the kind of original Virginia 94 strain, which is the first isolate of MG, because we figured that would give us the best sense of simply host evolution while kind of holding or removing any pathogen evolution that has occurred since. But we also used a more recent and higher virulent isolate to also just examine whether the effects we were seeing were strain specific or whether they're still kind of reproducible with more recent isolates. So for the analysis I'm showing you today, just for visualization, we categorized these 7 populations into 3 categories from like light blue, which included the populations that have the longest exposure. So Virginia, Iowa, Alabama. Then we have the slightly like the light purple and the dark purple here. And our predictions were that we would see if there was evolution of tolerance, that we would see the most tolerance in these populations that have had the longest sort of population level exposure to MG. And again, just to remind you, we measure tolerance using this slope-based approach. And here, we, as our kind of metric of fitness, we use I-score. So again, we're using this variation I-score here as a metric of sort of host tolerance, because now we're holding the pathogen constant and looking at variation in the host. And we know, importantly, we know that I-score over the course of infection is a good metric of the likelihood of mortality in the wild. So this is a reasonable proxy for the fitness costs of infection or tolerance. We used 2 different doses, particularly for our Virginia 94 strain, to try to get some variation in that x-axis. And we were basically trying to see whether we could see any kind of change using the space for time approach in the slope of this relationship across populations. So now I'm going to show you some data, and I've got the predictions at the top here, again, showing you like the color scheme. And on the x-axis here, I'm showing you the maximum pathogen load over the course of infection for all these individuals. On the y-axis is the maximum conjunctivitis score. And here I'm first showing you just data for the Virginia 1994 strain. And again, the populations go from like sort of least amount of population level time with MG in dark purple to the most amount of time with MG in light blue. Now the graph has a lot on it, so I'm going to build it a little bit stepwise. So first, I'm just going to show you the squares, and those are our pre-infection data. So we had control birds as well, but in this particular analysis I'm going to show you, we used every bird as its own control as well. So pre-infection, of course, we don't expect to see any conjunctivitis or pathogen load, and we don't. And you're going to, for each of these, see the individual birds kind of blurred behind the average data points. And there are actually error bars on here, but because no individuals had any load or score, you just can't see those error bars. But now I'm going to show you the data for post-infection, both of the low dose in the triangles and the high dose infections in the circles. And I know there's a lot going on here, so I just want to first direct your attention to the circles over on the right. These are the data for our high dose infections across birds from these different populations. And I think what's really striking here is that all of the birds from all of these different populations had almost identical maximum pathogen loads here on the x-axis. So all of these birds following experimental infection in a common environment end up with billions of copies of MG in their conjunctiva, but they show very different conjunctivitis responses. And it might not look terribly different, but these more tolerant populations, which represent Virginia, Iowa, and Alabama, actually have about half of the maximum conjunctivitis score than do these other populations that have less of a coevolutionary history with MG. We also see some really interesting results from the low dose, and I'm not going to have time to get into it, but we see some hints of resistance here where we're seeing a shift not only in the tolerance, which is the slope of the line, but also just in the average of the x-axis. So we see a shift to the left of maximum pathogen loads, suggesting that birds that are given low dose infections in these populations with a longer history of exposure have some higher resistance ability, but not so much at these high doses, which is really interesting in itself. Quickly, I just want to show you the other strain, that Virginia 2013 strain. We see really similar results for tolerance here. So again, we have these kind of less tolerant populations and these 3 more tolerant populations, which all have this longer history of exposure. So this tolerance result seems to be really robust regardless of strain. Now we only have 2 strains here, but it's really hard to do these experiments with wild birds. So we were surprised at just how robust this result was across the 2 strains that we looked at. And I should say here for Virginia 2013, we only used the low dose because for this virulent strain, we know that the infections kind of take off. As you can see, they got to the same kind of maximum pathogen load as did birds given a high dose of Virginia 94. So we concluded that House Finch populations rapidly evolved tolerance to MG infection. And this was really within the course of like a decade or 2. Now, of course, we cannot eliminate something like maternal effects, which may also be playing a role here. But we're pretty confident that there's at least some kind of genotypic contribution to what we're seeing here, even though there may also be some maternal effects at play that we cannot tease apart. We were also interested in potential immunological mechanisms. So I'm going to go quickly through this, but we did RNA-seq on a separate set of birds where we destructively sampled them at day 3. And for these birds, we only used the Virginia 94 high dose birds. So those were the circles that I boxed early on in those results. And we used those only, that treatment only, because we really wanted to specifically hone in on tolerance mechanisms. And that was the one treatment that really showed no hint of like resistance differences. So our hope was that we would kind of get closer to potential responses important for tolerance. And we only looked at the 2 extremes in terms of our population categories. So the 0 to 10 years and the 20 to 25 years. And we looked at 2 immune tissues. Today, I'm only going to talk about Harderian gland results. This is a secondary lymphoid tissue that's behind the eye. And so we suspect it might be important in a conjunctival infection. So I'm not going to get into any of the RNA-seq specifics, but we looked first at just like the number of total loci that were differentially expressed between infected and control individuals in each of these populations. And the one thing I want you to take away from this is that the birds from populations that are like less tolerant, which was Hawaii and Arizona, showed 10-fold more differentially expressed loci than did the birds from this kind of these more tolerant populations of Virginia and Iowa. There are also genes that were differentially expressed in all of the populations, meaning that infected birds, regardless of population, express these genes. But we were particularly interested in those that kind of fell outside that common area. We looked at the KEG pathways that these differentially expressed genes came from. And this is just to show you that they come from a diversity of pathways. It's not really surprising to see that they're largely from infectious disease pathways, immune pathways, signaling, et cetera. But the one kind of key result and the last result I want to show you is simply that when we dug into the genes that came from immune pathways, we were interested in whether tolerance whether tolerance might be associated with like the upregulation of various mechanisms in the system or the downregulation or neither. And we found across all of these kind of more specific immune pathways here on the left, that largely what we're seeing is that these less tolerant populations just have more immune upregulation early on in infection. So we looked at day 3 again. And so you see a lot of purple to the right here. These are just the number of genes that are upregulated. You don't see a lot of blue at all on the graph because largely the differences between these populations come from these less tolerant populations, basically upregulating more types of different immune pathways. We were also very interested in what types of pathways there are, and unsurprisingly, there are a lot of sort of innate inflammatory type pathways like cytokine interactions, but also a lot of adaptive type pathways as well. But overall, it looks like tolerance is almost associated with sort of a muting of upregulation of genes. So far, we think that there is some kind of dampening of inflammatory responses, but it's not that these more tolerant populations are really like specifically downregulating anything, but instead they're simply not upregulating these genes, at least at the time that we looked at, which was day 3. So the takeaway from this work is that that hosts can really rapidly evolve to minimize damage from pathogens. So we know that hosts can rapidly evolve in the face of infectious disease, but this rabbit system that's sort of the hallmark of rapid host evolution was perhaps not really representative of a natural system, given that we had this highly virulent strain that was introduced and kind of killed off 99% of the rabbits. But here we have an entirely kind of natural emergence event and a host evolutionary response that was quite striking and suggests that there was enough kind of natural variation in tolerance or in host responses for house finch populations to evolve over a decade or two. And this is really important because we tend to focus on evolution of resistance, perhaps because it's a lot easier to study, but we know that host tolerance has really important consequences for pathogen transmission and thus for evolution. And while we often think of pathogen as tolerant, while we often think of tolerance as causing sort of less selection for pathogen virulence, in this system in particular, given that we know that virulence may have these kind of direct benefits for spread in this system, we would expect this evolution of host tolerance to result in pretty strong counter selection from the pathogen to kind of be able to overcome this tolerance and still cause sufficient disease severity to result in that transmission benefit. So overall, we're really just kind of scratching the surface, even after several decades of work on this system. But we're hoping that by taking kind of host and pathogen and ultimately environmental perspectives, that we can start to shed light on the processes that favor the evolution of more harmful pathogens, both in wildlife systems, but also in human systems like SARS-CoV-2. So with that, I want to just acknowledge lots of collaborators and lab members, past and present, and our sources of funding. I'm sorry for going a bit long.
Wonderful, thanks, Dana. So I am fine to extend this. So anyone who is still here and has time, feel free to ask questions. Margaret, do you want to start by saying your question? Are you still here?
Yeah, sorry. So I was interested, why do you think that MG is more prevalent in non-breeding or low contact seasons? Is it related to temperature or other environmental factors?
Yeah, that's a great question. There are probably, well, we know there are multiple factors involved. So some of it is probably related to like the seasonal introduction of sort of hatchier birds that are first largely separated from adults, but then sort of become integrated into these flocks that are probably like a pulse of susceptibles. But there's also probably an important role for temperatures. So that's something a current grad student is working on. And she did a transmission study this past fall and did find that at colder ambient temperatures, there is higher transmission. And that's also probably multifaceted. So it's probably more birds are coming to feeders at cooler temperatures. They need to eat more. It could also relate to the survival of the pathogen on the feeder. So that's something she's going to look at specifically this spring, but probably lots of different factors involved. And yes, there are some cross-species infections. So goldfinches in particular are a key kind of spillover host. So the pathogen MG it can infect any kind of fringillid. So goldfinches, purple finches, crossbills are all susceptible, but it seems like the bird species that become infected are largely not important in like ongoing transmission. So house finches, by and large, seem to be the species that is contributing to transmission. That said, there are other species that do get infected from house finches.
Okay, awesome. Thank you so much.
Thank you, and I'm sorry again for going so long.
It's okay.
That's okay.
I didn't talk as quickly as I
We have
expected.
time. Dave has a question.
Hi, Dana. That was awesome. I really enjoyed your section on spreadability and symptoms that sort of have this effect independent of pathogen load. I was wondering if there are other symptoms that the birds might express that you think might be much more decoupled from pathogen load or transmission potential spreadability. Like, are there internal symptoms like swelling, changes in other phenotypes that might be related to virulence, but not related to like ability to spread? Maybe they're harder to measure or just more indirect or something.
Yeah, that's a really interesting question and not something I've ever directly thought about. I think there absolutely are. We've done some like histopathology looking at, what tissues is the pathogen getting into. And there is some, like, sometimes there's integration into the upper respiratory tract, but it's almost always limited really to the like kind of conjunctiva and nasal regions. I wouldn't be surprised if it's affecting like things like aerobic performance and sort of ability to fly, but we know that these birds are not moving around a lot on the landscape.
Yeah.
I tend to go to behavioral effects, but I'm sure there's also potentially effects on the diet that they're choosing. So that's work that someone in Jim Edelman's lab is now looking at, like foraging decisions, and whether those change when birds are infected. So it'd be really interesting to see if, for example, birds aren't thought to use their sense of taste much for foraging decisions, but there certainly could be changes in the extent to which birds rely on feeders at all, or choose particular diets when they're infected.
Yeah, I guess maybe this is like COVID bias coming in, but I was wondering, like, do you think that the symptom kind of profile could be changing? If you're sort of attributing things to tolerance, it's like, if you're measuring one sort of disease phenotype but not others, maybe it's like a shift in composition of symptoms.
Oh, like something's happening internally that we're not.
Yeah, I don't know.
That's interesting. We haven't really quantified this, but we also did at least do this sort of qualitative description of the different types of visible pathology. So,
Cool.
And we did note some interesting differences among the populations that look like so sometimes you just see like what we call eversion, where it's like, it's not really swelling, but the inner conjunctiva kind of pops out. And so we haven't really explored those data very well because they're a lot harder to kind of analyze. But yeah, that is something they've seen in the myxoma system, where like the whole pathology is really changed of infection. So yeah, that's a really good point. Is we should look at whether there's something else going on that we're not seeing.
Cool.
Thanks, Kelsey.
Thanks.
So I love the seminars. You get these great questions that then show you what you need to do.
I have another question, and that's, the system has been such a great model system of this emerging infectious disease that's tracked over time with all those different theories you can test, right? And so when you think about the literature, there's multiple ways that would drive virulence upwards. So with and host competition is one, and you have looked at that in several of your papers together with Andre as well. Then, of course, there is the idea of resistance or tolerance being able to drive virulence upwards as well. And I was just wondering, looking at your data with the tolerance, what's really interesting in those graphs that you showed is that there is some signal that resistance is greater for the birds that have experienced the outbreak for, about 20 years compared to the ones that have had it more recently. But the bigger difference, like you pointed out, is in tolerance. And I wonder if you have any feeling for whether that has gone hand in hand, as whether that increase in tolerance has also facilitated that greater increase in virulence that you showed.
Oh, so whether resistance and tolerance went hand in hand, but whether
the tolerance and the virulence increase have gone hand in hand, because that's certainly what we predict with simple tolerance models, right?
Right, exactly. Yep. So I think one thing that's interesting about this system is the virulence increases start well before we see any hints of resistance or tolerance. So we had done some earlier studies looking at potential differences in host populations and didn't find any signal. There are other groups started to see differences in resistance popping up around, I think, 2007 maybe, but I'm not going to pull back up my slides, but the increases in virulence actually start before the hosts start to evolve, like within like a couple of years of the pathogen sort of emerging on each coast. So we interpret that that's been sort of the driver of a lot of our other work on that I didn't talk about today on like immunity and how sort of prior exposure can favor like standing immunity that birds have can kind of favor more virulent strains. So I certainly think that the evolution of host tolerance is likely now contributing to virulence, but I don't think that explains the early, like the start of it, if that makes sense. But there are probably so many factors that are contributing that we need a really good model to really try to tease them all apart.
Yeah, no, I think that makes sense. So it's almost like you have this jump in species, and then maybe some factors drive that virulence upward. Maybe it's greater transmission opportunities, so more horizontal transmission opportunities, maybe more within host competition. But then once it's more virulent, the host evolves more tolerance. But then you could potentially predict right now that that's going to drive virulence
Even
up,
higher.
right? Because the theoretical idea is that you're more tolerant, so you suffer less from the consequences. The cost in terms of the trade-off for the parasite is less, so it can evolve even higher virulence. So,
yeah, exactly.
cool.
Yep. Yeah. So I think we have a lot of work to do in terms of where would those predictions go.
Yeah. Well, I think the nice thing about studying an ongoing outbreak is it provides some career security, right?
That's true, especially in light of the SARS-CoV-2 pandemic, for better or worse. There's certainly a lot of interest in infectious disease dynamics. So I think we all have career security there, sadly.
Yeah, but it's always nice to focus on a completely different system, you know, and
absolutely, yeah.
It allows you to look at things differently.
Yeah.
Because one thing you don't have to worry about is all these interventions, right? And this is a real wildlife disease. There are not many examples where we have these long-term data sets on wildlife diseases that really allow you to look at the evolutionary processes, not just the ecological process. And I think that's one thing that's always struck me about your system as a major benefit that very few systems have.
Yes, exactly.
Yeah, great. So super.
Yeah. Thanks. Yeah.
All right. So we're gonna hang on here, but I want to thank everyone else for being here.
And I'll see some of you later. So thanks very much.
Thanks again, Dana.